to the Weekly Bioanalysis, a KCAS Bio podcast.
Hello, and welcome to the 95th episode of the Weekly Bioanalysis, the official podcast of KCAS Bio. KCAS Bio is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 45 years now. My name is Dominic Guarino. I have been in the LBA and biomarker space for a little over 30 years. I'm here with my co-host, John Perkins.
Hi, I'm John Perkins, and I've also been in the bioanalytical space for a little over 30 years.
John and I are members of the growing scientific services team here at KCAS Bio. Either or both of us are available to answer any of your questions you may have regarding this podcast or any of KCAS Bio services. We are amped up to have you listening to the 95th episode of the podcast, which is now available virtually everywhere. Wherever you choose to find and play your podcasts, you can now likely find the Weekly Bioanalysis. We welcome all of you, whether you are joining us for the first time or if you're a regular listener. Today's podcast will be a review of the latest news and resources, and then a focus on a topic of our choosing before discussing any feedback we've had from you. We are constantly looking for topics, and we'd be happy to discuss something that you want us to cover. To kick us off for podcast 95, John is going to go over today's podcast topics a bit before we jump into the news and resources section. John?
Yes, so today, as usual, we'll go through some news items that we found that we thought would be interesting to discuss, and then the main item today is really talking about the highlights and impressions from both the AAPS main meeting and then the EBF Open Symposium, as we've both been travelling, and obviously it's a chance to connect with other scientists and colleagues and learn a bit more what's going on in the industry, etc. It's nice to have a recap on what we picked up during our travels.
Great, John. Well, let's just jump right into it. What's our first news article?
Our first one is a Nature article, powerful new antibiotic that can kill superbugs discovered in soil bacteria. And this is part of the ongoing quest to try and find drugs that can overcome the resistance of microbes to actually tackle some of these very stubborn infectious diseases. And this was a group at University of Warwick in the UK, and they were just looking at the pathway to make methyllinomycin A, which is a well-known antibiotic. And in looking at the pathway, they discovered a couple of intermediates that were substantially more powerful than methyllinomycin A, and it was also shown that it could kill bacteria that were the underlying cause for some hard-to-treat infections. So it's like they were a little bit astounded by that, because as they said, they anticipate that evolution perfects the end products. You'd expect the final molecule to be the best antibiotic and the intermediates to be less potent, but this was not the case with this, and it's a big problem. I mean, antimicrobial resistance is expected to cause 39 million deaths worldwide over the next 25 years. So this is not insignificant. And as we've talked about in the past, for pharma companies, it's a difficult thing to get an antibiotic to market and make money from it. So although there's the need, it's not really a high priority for pharmaceutical companies. So I'll pass this on back to you.
Yeah, it's interesting. We don't hear quite as much about some of this, but still hasn't gone away, has it, John? There's been no major advancement there. But, MERS was, I think people are, I wonder if like COVID had like an effect on people being clean, although I start to see a lot less of that these days. John, I guess I have a question for you. This is right up your kind of alleyway. Why is the intermediate? Can you like kind of elaborate on why the finding that the intermediate compound that was 100 times stronger than the final product? Does that mean the final product will be 1,000 times? Like, why is that such an important feature, I guess?
Well, it's basically saying that methyllinomycin A is the antibiotic that the bacterium Streptomyces colicolor synthesizes, but there's obviously a bunch of intermediate compounds. I don't know what it starts with.
I see.
And so it goes through various intermediates to get to that
Mm.
methylomycin A. And this, like I say, this was their thought. It's like, well, if this is what the bacterium's come up with, although quite why the bacterium think this is the boy that's going to make all the difference. They were thinking that the bacterium has obviously developed this to counter something that it deals with.
Okay.
And so their thought was, well, if that's the bacterium's final product, then that's going to be the most potent one. But as they actually tested some of these, it was actually a couple of intermediates. They said there's the pre-methylamycin C-lactone is much more effective at targeting 7 strains of bacteria, including Staphylococcus aureus and Enterococcus faecium. So Staphylococcus aureus can infect skin, blood, and internal organs, and Enterococcus faecium, which can cause bloodstream and urinary infections. And I think there was actually another intermediate as well that also showed more activity. But it was saying that this they're a mouthful. Pre-methylamycin C-lactone used could kill Staphylococcus aureus with just 1 milligram per milliliter compared to 256 milligrams per microgram per milliliter compared to 256 micrograms per milliliter of the methylamycin A. And so then their next thought was, well, how does this compare to vancomycin, which is our current last line antibiotic, to see if resistance is an issue with this. And actually, from what they did, they I think this was all in dish. It'll be all in dishes, but they basically the vancomycin treated bacteria mutated and developed resistance for the strain that they were looking at, which was the Enterococcus faecium. But the amount of pre-methylamycin C-lactone that was effective was fine throughout their study, and it showed that Enterococcus faecium didn't develop resistance to it. So it's one of those things that it's showing a principle, and obviously it's very early in the research process, but they want to now check is this molecule toxic to mammals, and then does this give a path forward? And then are there pathways where they could make analogues that if they discover toxicity with the pre-methylamycin C-lactone, can they make similar compounds that retain the activity but don't have the same toxicity? So, like I say, early studies, but it's just a really nice example of, even though the research, they talk about this, the methylamycin A was first identified in 1965. They started working on this in 2006. They mapped the mechanism of synthesis in 2010, and it wasn't until 2017 that they started to dig into the intermediates and found that there was this pre-methylamycin C-lactone, which was more potent. So it's indicative of old research can still point.
Yeah, it's fascinating.
That's why it's an example.
Yeah, no, it's really interesting. You just, that last point about it being discovered in 1965, and here we are, I don't know, 90 years later.
60 years later.
Seeing that there's, they had this intermediate they didn't even know about, right, John? That's kind of what the story goes. Yeah. And it's, like you said, it's still early stages. I think it is indeed in a dish or a petri dish to doing all this, but looks really promising. And I don't know, maybe I'll sleep a little better tonight knowing that somebody's out there in the good old UK working on stuff that'll prevent us from having microbials overrun us, which I think was a fear not too long ago.
Well,
so stuff.
Yeah, I think drug resistance is always a concern, absolutely. Yeah. So let's move on. Next one is another old global issue. But it's still fascinating because of this. I mean, we'd like to talk about what's cutting edge, but ultimately some of this other stuff that hasn't been resolved is just as interesting. And this is basically experiments revealed targets for new tuberculosis vaccines. So this was a guy at Samuel Bihar, a tuberculosis researcher at the UMass Chan Medical School. Oh no, sorry, I've got that wrong. This was actually, it was started by Brian Bryson, a bioengineer at Massachusetts Institute of Technology. It's the Samuel Bihar was commenting on the study. Sorry about that. So basically, Bryson's work revealed that cellular signatures that signify tuberculosis infection yielded a series of targets for new vaccines, and hopefully give a path forward for addressing TB. And TB is, again, we're still in the bacterium field. It's caused by Mycobacterium tuberculosis, and it kills about 4,000 people globally every single day. That's actually, I read that a year earlier, and so it's a day. Wow, that's amazing, isn't it? But so far we only have one vaccine, and that's the Bacillus Calmette-Guerin vaccine, the BCG. That certainly those of us who grew up in the UK in the 70s all lined up for a BCG vaccine, and it was one of those ones. It didn't heal. It might, depending on who you were, it didn't necessarily heal very well. So mine took months to heal, and if you ever like brushed on it or broke the top off, it was always painful as well. So it took, and we all have like our BCG scar on our arm from where the injection site was. Anyway, that was first come up with in 1921, and it's basically just a weakened form of a related bacteria that infects cattle. And according to the article, it says BCG prevents severe forms in TB in children, isn't effective in all populations, and the shot doesn't work at all well in adults. So there's a lot of scope to do something here, and this is really nice because it actually calls on mass spec helped out with all this. And
I know, John, it's funny, before you dive in, I was like, oh, I know where they're going with this. The only time I ran a mass spec is about ready in grad school, is about ready what you're going to go over, but sorry, go ahead. Yeah.
All right, that's fine. It's great. So basically, so I'll talk about so when the cell is infected with the Mycobacterium tuberculosis, the cell, as part of its defense, chops up the bacterium's proteins and presents them on the surface as a kind of alarm signal to the immune system that something's going on. So Bryson's team infected a variety of human cells with TB and then harvested the receptors from the surfaces, and these that then present proteins to passing T cells, and these by harvesting the receptors, these are called human leukocyte antigen complexes, and they were looking at these complexes from these cells with mass spec. Yeehaw! And they found that many infected cells were presenting proteins used by the bacteria to acquire nutrients. As part of a system called the Type 7 secretion system. So this is way over my head, but I like to say, he actually said, so they worked on identifying some of the tuberculosis antigens sitting on the infected cell surface alongside, and this is like a needle in a haystack thing. There's tens of thousands of other proteins on the surface here, but they're going after the key ones. And ultimately, it was the advances in mass spectrometry that actually made this possible, so it's we talk about this on the podcast a lot about, from where we were when I started doing mass spec, 30, 35 years ago, and where we are now is night and day, and in terms of what the technology can do, and just the extent of the molecules that we can look at, or some of the complexes, etc. So this is really interesting from an analytical chemistry point of view as well as a medical one. I actually ended up at the booth at EBF, and I know this is part of the main topic later, but I ended up talking to Guy about how old the interfaces were we'd used and things like that. See, when you get like old mass spec people together, they start getting all nostalgic. Anyway, so I'll pass it to you on this, and then I might chime in a bit more.
Yeah, no. So, what the mechanism of how TB is, why we can't eradicate it, right, John? Let me start there.
Yeah.
So it's real clever when your antigen presenting cells take it up, and most of the time bacteria gets taken up by your APCs. Those are like B cells, dendritic cells, and they bunch it up and they present it in these HLA complexes, and they present them in HLA-1 that triggers a CD8 response, a killing response, and then they present them in HLA class 2, and those are humoral antibody responses. And so the bug has a way of altering. So TB gets taken up, and it can either, if memory serves me right, there's two things it does. It doesn't. It somehow can put like when it presents its antigens. So when normally you present these antigens and it would signal the other immune cells around you to come and kill the cell or generate an immune response. It does it in such a way that it almost immediately, instead of going into like a pro-inflammatory response, it goes into a suppressor T cell response. You see that, John? So it doesn't allow for any sort of acute inflammation to go and kill the cell. So it's able to evade the immune system with the type of antigens that it's presenting in its HLA. That's I think the major. And then they have ways of becoming. So a lot of things that trigger a more pro-inflammatory response are things like IL-2 and IL-12 and even IL-23, and it kind of ablates them as well. It's got like a dual mechanism of putting up the way it gets processed within the TB itself gets processed, the antigen processes, the way the peptides are processed and presented. And that's what they do mass spec for, by the way, John.
Yeah, absolutely.
You use an acidification step and you take out all the,
Yeah.
and like I mentioned earlier, that's the first thing I did. So the bug has a way of naturally kind of resisting. And what the really fascinating thing here, and then I'll pass it back to you, is it looks like they used mRNA vaccine.
You did, yeah.
Not instead of like the more traditional way is to look at, they're introducing new variables of the bug, right? So like everybody presents the same sort of proteins, and that's why I kind of went on that. Like what I was trying to explain. I don't know if I did a great job because I'm going back years.
No, understood. Yeah.
There was a lot of groups that studied TB. I haven't researched it in a good 20 years, but when those everybody presents the same TB proteins,
Actually, that's
how
what
it evolved, right? That's how it sat there and said, okay, instead of I now know how to take anyone's HLA type because we're pretty diverse, and instead of just causing this pro-inflammatory response, I'm going to suppress T cells and I'm going to live, and then I'm going to kill you over time, right? So now they're introducing what, right? They're kind of introducing new TB proteins, right? Is that the way this?
So yeah, but according to what was said, not everyone does present the same. Not everyone's HLA complexes do present the same proteins, so they're still focused on identifying the diversity of what might be expressed. But that this was the mRNA approach. Basically, found that the cells were able to use mRNA to build the type 7 secretion system proteins.
My bad.
Yeah, no, that's fine. I'm just saying because, like I say, it's a fascinating article, and so trying to get my head around it is.
I think I'm still half right though, because they are so sorry, I had that completely wrong. They are introducing. So you're right. Well, everybody's got a diverse HLA type, but they are somehow introducing this type 7, right? So they're like forcing the immune response into something that could ablate the TB. It's early, John. I'm going to cut myself a break. It's okay. It's all good, but it's a great story, and it brought me way back into my early days of my PhD in immunology there. And I just remember looking at the mass spec people going, "Oh my God, this is so boring."
Okay.
No, because we were using like we had to strip off these cells, and it's part of how we started discovering HPV peptides and things like that. That's what
I think it's one of those things that with something like this, once you find a path where it's starting to give a hint of what's going on, that's when it becomes exciting. Yes, it can be a lot of legwork to try and dig into this, and that's obviously what these people did. Was, it was like they must have generated so much data to then sort of start zeroing in on the proteins that were part of the process to then go from there. So ultimately, their aim is to come up with a vaccine with this approach and hopefully do some, address what is a chronic health issue globally. So yeah,
it's fascinating, John, though, because you're right. It is a needle in a haystack approach. It's really impressive because people have been researching these types of HLA peptides, because that's ultimately what you're pulling from the mass spec is a peptide. They keep saying protein here, but they've been researching these things for decades, and to find one that could be a kind of a pan vaccine is wonderful news. And TB is still a huge problem, right, John? It's something that
sure, yeah. They said 4,000 deaths a day. I mean, I think it's with something. If they are proteins, yes, you're going to digest them. Then
is it 4,000 a day? Sorry,
that's 4,000 a day. That's what it said in the article. Yeah, that's a lot of people. If these are proteins, you're going to be probably digesting and then piecing the protein back together to get a sequence and identify it on the basis of that. So I don't know what size they are. Obviously, it doesn't go into that kind of.
Memory serves me right. The HLA class 1 is a 9 to 12 peptide, and the class 2 is anywhere from 10 to 13.
Yeah, but even then, they sort of said somewhere they're working on one protein in the vaccine for the last 6 months. So some of these proteins are a nightmare to purify. So it's not just there's all the purification and all sorts. So yeah, this is like a gargantuan task they've set themselves, and good luck to them. And
4,000 people a day is horrific. And man, I when we get one TB death here in the U.S., I think it's major news, right? We don't.
Yeah, exactly. Yes. So let's move on.
Yeah.
Final news item. UBC enzyme technology clears first test towards universal donor organs for transplantation. So this doesn't really impact us. Well, it doesn't impact us immediately, but it's just, again, a very interesting example of the way people think. So the first successful human transplant of a kidney converter from blood type A to universal type O used enzymes developed at the University of British Columbia to help prevent a mismatch and rejection of the organ. So this was in Nature Biomedical Engineering, and it might be a step to where kidney transplants become much easier to basically help patients out with kidney issues. They had this. So the underpinning piece of it is this is a guy called Dr. Stephen Withers and his colleague, Dr. I hope I don't mess his name. Dr. Jayachandran Kazakidatu is a professor in the Department of Pathology and Lab Medicine in the Centre for Blood Research. And they worked on stripping away the sugars, enzymatically, the sugars that define blood types. So we've seen things about transplantation of organs, and then the follow up that you have to do to ensure that there's no rejection long term, etc. So basically, the mechanism is these sugars coat organ blood vessels. If a recipient's immune system detects the wrong antigens it attacks, type O patients, more than half of kidney wait lists, can only receive type O organs, yet type O kidneys are often given to others because they're universally compatible. As a result, type O patients typically wait 2 to 4 years longer, and many die waiting. So like I say, by stripping these sugars off, they can basically take kidneys from a wider population and make them more available to the population that are then waiting. I think they don't mention what the enzymes are. They just say they're highly active, highly selective, and work at very low concentrations. And the proof of concept was done. They had an enzyme converted kidney that was transplanted into a brain dead recipient with consent from the family, so that the researchers could observe the immune response without her life being risked. And so the kidney functioned for 2 days without hyperacute rejection, and then by the third day there were some blood type markers reappeared, signifying a mild reaction, but the damage was far less severe than a typical mismatch, such that they think this could be a long term. This could be a useful way forward for tissue transplants and how to overcome that incompatibility. That is obviously an issue for tissue transplants.
Yeah, no, fascinating stuff, John. In terms of, it's really cagey to say they just used enzymes. They don't really talk about what they did. I could see why, but we hear about type O blood types, right, and A, B, and the universal donor, and I always get this mixed up as type O, right? And so I think I think they leverage that kind of knowledge from things, and there's a lot. It's again, a field that's people have been trying to put organs into transplantation's been extensive over the last 30 years, maybe longer. But certainly there are many things that they try to perfuse the organ to remove all the antibodies, all the cells. They've looked at suppressing it with meds. But this is the first I've heard of hey, why don't we, they kind of talk about stripping away the red paint and getting back to the neutral color. So it's you can take a type A, type B, AB person, strip away all those sugars and antigens, right? Those kind of blood factors. And now you got a type O organ. Yep. Well, heck, how did it take so long to figure that out, John? On the surface, that sounds easy, but it's
on the surface. That's the key response, isn't it? Yeah. On the surface, it sounds easy, but yeah.
No, but that's it's such a. God, that how so. It's fascinating the iterative approach it sometimes takes, and then when you look at the end product, you're obviously oh wow, why didn't I think of that? No, yep. John. See, that's why I'm sitting here with you. No, but it's a really great story and really optimistic with, because they talk about what there's much more than just kidney, right? They start talking about lung and other organs. Yeah. Be a universal. It's a little morbid to think they put it into a person in a coma, right, to make sure it's okay, or a brain dead individual or something. But it's at the end of the day, these are the types of things we have to do to ensure things are right. But this could be game changing. The field had advanced in quite a bit with put human organs in pigs, right? That's where
absolutely, yeah.
I think the future had been lying in. We could grow pigs with human hearts, and we could all get new hearts whenever we needed them. And I think that's come a long way. They've overcome a lot of this. I don't know about the matching of the sugar types, but a lot of it had to do with complement. I still think there's hurdles here. Maybe that's where I'm going. There's other factors that, although this is a major one, without a doubt, if you could remove this huge hurdle, the other ones with graft versus host disease is probably more overcomable with some sort of medication. But this major mismatch, right, if you're not type O to sort of stripping away, but there's not that I want to bring a bad, negative light on here. There's still work to be done, but this is unbelievable, John. This could be our grandchildren could never be on a waiting list for an organ transplant if they could figure this out.
Sure. So, just when you say, Nick, there's work to be done, so they're looking for regulatory approval for clinical trials, and then they have a spin-off company partnering with UBC. They're called Weekly Bioanalysis, which will lead development of these enzymes for transplant application. And to enable the creation of universal donor blood on demand for transfusion medicines. They're actually thinking outside of organs. They're actually thinking blood as well. So, yeah, there could be a lot here if they get this right.
Yeah, no, that would be that's another. I guess I kind of missed that in there, but yeah,
that's right. At the tail
blood
end.
drives, right? Blood drives would be you would never need you'd still need blood drives, but there would never be a shortage, right? Because if you got all these ABs and you need Os, that goes away, right?
Sure. Yeah,
and then a major implication for any sort of soldier out there who might need help in the field as well. Unbelievable stuff. Really cool.
Okay. Yep. Okay.
You're picking me up, Jonathan. Let's
move on.
Yeah.
Yes. So, main topic. So we've obviously been travelling for the last few weeks, and some of us are still in the UK, getting ready to fly back tomorrow. But as we did a sort of precursor to the AAPS and EBF Open Symposiums a month ago, we thought it actually made sense to talk about what we'd seen and what came out of those meetings in terms of highlights and impressions. And I mean, I don't know. Do you want to start and give your overall takeaway
Yeah,
from
sure.
AAPS just to get things going, and I'll
Yeah.
chime in with some of my EBF stuff.
Yeah. So, well, unfortunately, I was unable to arrive until Monday due to a personal event on Sunday. But I didn't get to go to as many talks as I would have liked to had. But I certainly got the vibe, if you know what I mean. There were three major things that, at least in the world I was swimming in, John, right? Because AAPS
Sure.
is, I don't know, it's maybe 3,000 or 4,000 people. It's not as big as it once was, but there is a lot of CMC and, a fair amount of like pharmacists and students. But there's always a nice bioanalytical presence. And actually, I guess it's four topics. The first was AI, right, John? That kind of
Sure.
probably
Absolutely.
dominated
Yep.
everything.
Yep.
And I don't need to go into too much about that because it's still in play, in my opinion. We're still, there's people who are actually starting to maybe, use it to analyze their data. I still think that has some risk to it, but maybe you have some insight. But I'll move
I'll
on to
say
my
a little
second
thing.
one. Yeah, I just to me it's just still hard to trust it, hard to not need someone to QA it,
Yeah. Yeah.
but I'll move on to the second thing,
So,
and
so
that was
why don't I jump in on that because
sure.
I had it as my list on my list of things to talk about with EBF, and it's fine. So yeah, it for for the EBF, it was one of the primary topics that within the meeting AI, and I think it's my impression was that it's a drive within large pharma. I sit there as an audience member, not really knowing how it immediately applies to us as a CRO. But I mean, and also because so EBF is it obviously has a lot of parallel sessions. So it was one of the it was highlighted as a key topic right at the beginning on the first day. But then because of parallel sessions, I didn't go to the AI session, but there was a 4 hour workshop on it on the afternoon of that first day. So there's obviously a fair amount of discussion going on about it within EBF, but that's really all I have to do. Have to
Well,
say.
what I'll add is a lot of people, like in some of the circles I was running in, they said they just didn't learn anything new.
Sure,
That,
yeah,
that was kind of what I was told. Although I don't know, like I said, AI, we're all going to use it. It's all good. I mean, one could make a case, Google searching and things like that. We've been using AI for a long time, in my opinion, but it is coming to a whole nother world. And the problem with it is this personality concept, because John, I don't want to get too philosophical, but we're all innately evil in some capacity, right? And so that makes it really rogue unless you start practicing religion. I don't know, or AI goes to church. I don't know how it would help it. I don't know.
yeah, yeah.
But I'm not going down that path, right? So the second thing that there's the second one for me was I got a chance to, I got invited to be a member of the biomarker community, and they had a big dinner. There was 17 of us at that dinner, and that was really enjoyable. And the biggest, still the hot topic, and maybe I don't know if this really came up at EBF. They might be past it, but it was this qualification versus validation
Oh
concept.
yes, oh yes, oh yes.
And so this is one where I still think it's clear as mud in my mind. I mean that it's really we just. I think you've heard me maybe talk about this before, but I think I feel like part of the reason why they invited me, I gave a talk on this at an e-talk, and really what the confusion to me comes twofold. One, it's this people clearly are not defining what they're saying. It's not a it's you have to be careful. Is it clinically validated or
Mhm.
analytically validated? And that to me is one factor, but there's a second piece here. And that's in the CMC, the GMP space. When you say a method's qualified, that means it failed validation and it gets a QA review. John, that does not mean that to you and I.
No, no, not at all.
Qualified method could have
No.
less than 5% relative error, and but it's still, by definition in our world, it's not getting a QA review. So that was part of what we just, kind of, that's been my shtick lately. And in part, I want to thank my wife who works in that space. But I was talking about it, she's like, no, qualified methods fail validation and they get a QA review. I'm like, holy crap, that's the problem. So I think that's where you got a couple worlds colliding, right? You got like the physician's world, you got the CMC world, you got the GMP world, and they're all colliding. But I'll kick it back to you before I go to my next.
Yeah. So I think it was less on the confusion between qualification and validation. I don't recall too much mention of qualification, but it's really it was sort of, there was the EBF stance was, we talked about context of use validation. So if we apply context of use, for like secondary matrices, things like that, then we should be able to get away with much less validation. And I think there was some agreement in the field, but the EBF sort of detects a backslide, and we're going back to that more conservative position of if we have a matrix, we should validate it. And so they were really talking about, again, tying into that, there's the 3 R's where, where they're talking replacement, reduction, refinement. We're trying not to consume materials or matrices such that has an impact on animals. So it's all part of that. It's so for things like they basically said biomarkers, urine, tissues, you do not need this full validation. And I think it came down to some extent some confusion with ICHM10. I think I think there, from EBF's perspective, having been really pushing for context of use to try and minimize the validation experience and having validations being scientifically driven, they want to get more back to that and actually bring that more as a focus for wider discussion. So that was more the discussion.
Yeah. Well, I'm still confused by what you just said, because like, what's the harm in saying it's and one thing that we did talk about as well was these tiers, right? So,
Sure.
but like, you still have to call it something, right?
Yeah.
Like if you just say it's a tier 1 validation, tier 2 validation,
Sure.
tier 3 validation, tier 4, and that has meaning, that's fine. But really, does it matter if you call it a qualification? I don't know. And
like I said, I don't think qualification is really mentioned as that might be more.
Because they just stopped using the word. I think that's why it's not if I've been told if you say the word qualification, you're gonna really be off-putting in a biomarker context.
I think from the EBF perspective, it was we want to be sure that we're not always going this conservative approach of doing full validations all the time and trying to put a break on that. So you're basically not doing unnecessary work, and so it's like they have like I've got screenshots of the what you do. Obviously, primary matrix like plasma, that's your full validation, which it might vary depending on the application of the drug. But things like urine, they had like here's the experiments you could drop, and we never do. I mean, we when we're doing small molecule validations right now, we're quoting full validations for plasma and urine, and maybe that's something we should revisit.
That's really for PK, not necessarily biomarker, right?
No, that's for PK, yeah, absolutely. Yeah. But again, we've had we will get customers to say although it's a biomarker and it's not a primary endpoint, we still want a full validation, and it's like, well, why, So anyway, that was just part of their discussion. So yeah, you can. What was your third thing?
Yeah, no, we could talk a whole time about this biomarker.
Sure, absolutely.
You can almost sense you kind of struck a nerve with me in there, didn't you? So it's so funny. The third one was immunogenicity. So there's a lot of push to eliminate the 3-tier approach and move it towards signal to noise. And, there was a fair amount of examples, but I don't, I'm warming up to it, John, but I feel like I want to throw some caution at this just from it's something where I think you don't want to be in phase 3 and be like, oh man, really, we should have done a 3-tier validation here. So but clearly to your 3R point, I have no issue with significantly reducing the, and
Mhm.
we already are, although the regs haven't changed yet, John. Remember,
Sure.
you're at, to some degree, I want any audience listener to know that you're at risk unless you've kind of worked with your FDA filing the right way, without doing a 3-tier validation for your tox studies. Although it's becoming more and more the norm to do a lean approach, or even just, and a lean would be like a 2-tier validation, or even maybe a single combined, like screen and titer at the same time, but it's still a validation. Some are doing, I'm going to use the word qualification here, or even using the signal to noise approach, but that's a risk, John. And the regulations do not necessarily haven't changed yet. And then when you get into the clinic, so for me, I when it comes preclinically, I think it depends on the assessment of the drug, but I don't think you could just say I'm not doing any immunogenicity in a biologic state.
Sure.
If you're
Yeah.
using a monoclonal antibody, I think a rough and ready method's probably good enough, unless you see an animal's kind of keel over on you, then you might want to do more. But then you get into the ADC space, I really think, or some sort of more high risk, drugs. I do think you should have something, but maybe not a validation, but I do think you have to look at some development of the method that gives you some assurances that you're seeing, more than just a signal to noise approach. Although again, I think I'm old school, and maybe as more data, maybe you show, hey, I got during development, we can do these 3 tiers, but hey, we're only going to validate a signal to noise approach because we're not seeing a lot of risk. Something like that, right, John? There's just like some common ground. And then when you get into the clinic, I think you really, in the phase 1 portion of things, I want to be a little more conservative. But as you get into later phases, I got no problems with,
Mhm.
after our first in human study, moving solely to signal to noise. And you want to talk about eliminating a whole bunch of costs and unnecessary things. It's that neutralizing antibody step, which is probably the next thing that they're really going to show. I don't think they've quite shown that data, but there's been consortiums on screening and confirming, showing that you could really, a high percentage of the time. Although we've done some retrospective stuff specific to ADCs, and we're like 6 or 7 out of 10. So there's still that upwards of 30 to 40% that the 3-tier would have been applicable.
Yeah.
John, I'll pass it back to you, but
Yeah.
I don't know if there's a hot topic there, but anyhow.
Yeah, so again, this is outside my sphere. But while I was looking through the program, sort of because I knew this was at least one. There's one session that touched on the title of the session was 1-tier technology, single tier prime time, and it was presentations from Synexa, LabCorp, Charles River, Bioanalytics, and Sanquin, all talking on this subject. So it's obviously a hot topic within the EU as well as the US. The other interesting one, and I do want to shout out to Blondine Dumont from Lyon, was there actually a workshop on ADA for ligos and peptides, and I know that we've been in calls fairly recently where we've been asked to quote on ADA for peptides that you'd ordinarily think wouldn't be an issue, but it does seem like there's increasing focus on that for the moment. I was not in that workshop; I was catching up on other stuff. But I know there were other people from the company were in there. But certainly, Blondine's presentation got a lot of questions, and was there was a lot of discussion on that.
Yeah. Likewise, the ring trial, is that what you're referring to, or is that something different here?
I honestly don't know. I'm just sort of passing on what I heard.
Yeah. So the ring trial is an oligo trial, and we are participating in on the PCR piece. Um, for unfortunately, we're there. The other things, we're just a little late getting to it, but Dawn attended some of that, and I heard through her some interesting updates on just comparing platforms. That's what this is looking at. It's an extensive look at comparing, whether it's a mass spec method, a hybrid ELISA method, PCR method, or even a hybrid LCMS method. And I think that the answer here is still to be determined, but
Mm-hmm.
it's really I think it's not only is it a wonderful scientifically to assess oligos who are, man, they're on the rise, right, John? We all know oligos are coming, and so it's a really powerful scientific study to help with just the development of it. But I think it shows some collaboration that was kind of waned since COVID a little bit. I think that this is a good one to me. Remember, during COVID times, it seems like we were all kumbaya, and then slowly we kind of started getting a little more kind of not against each other, but a little more tight-lipped. And this is a good collaboration amongst everybody.
Yeah, it was interesting that Legos are obviously on everyone's mind, but yeah, and it's obviously like when you're compiling a program for a meeting, you're trying to find something that meets most people's needs. Legos were in talks, but there was no specific sessions for Legos this time around.
Oh, so what was?
So I'll touch on that, but I mean, but so there was at least one talk I went to. There may be more. I can't remember offhand. But yeah, it wasn't like they were being ignored. They just weren't at the forefront of what were being discussed.
I guess I missed it. What was the
workshop was on ADA for pet.
Oh, got it. Man, I got way off there. Yeah.
Like we've been in calls where we're looking at.
Oh, that was our colleague did that. Yeah. No. Sorry, that was a great. That's really exciting.
Yeah. So anyway, yes. So your fourth point that you had.
So they're overhauling AAPS community.
Okay.
And this has caused a lot of concern. So John, I don't know how much you know about or even our audience knows about the AAPS focus groups or the community in general. I think there are 32 or 33 community focus groups, and one of them is the critical reagent one that I co-lead.
Yep.
There's PK. There's a whole bunch of them out there, and I think I'm active in 3 or 4 of them. Lead one of them, and so they are eliminating all of them and bucketing it into 6 communities only. It's going from 34 actually, and not only that, that's just in the. So to me, I'm still trying to really fully wrap my head around this because I hear different versions from different people. Okay, so there's a lot of hubbub. I've sat in on some of the community leadership meetings, and there's a lot of pushback because you got people that have been doing a PK group for, I don't know, I've been on that for 9 or 10 years now, and they've been that individual's been running it for I think 12, and you've got people that rely on this sort of thing, and that's all going to be rolled up into a single community where, and I should have pulled the list up, but there are 6 different communities now, and so a lot of it, but those communities are actually, some of them are going to be environmental science and CMC, GMP. That's not going to be applicable. So the 34, the 32 groups down to 6 is really causing some headaches for people to try and figure out hey, if I'm in the PK group and the biomarker group, but now there is a bioanalytical group and a translational medicine one. And so people can't figure out which one they're going to. And the shorter answer is you can go to both. Yeah. But then the number of leadership opportunities is being reduced dramatically.
Sure.
To me, I roll with it, John. It doesn't bother me. I'll find out one of the best things about the AAPS community is it allows you to find answers or find right people. It's just a great networking thing. It's good to give, it's volunteer time too, John, right?
Sure.
And so it feels good to give back to, there's a lot of young people on there looking for answers, and just, even if it's just career development, whatever it might be. It's kind of fulfilling a little bit. And so for me, I'm at a stage in my life where I don't. It doesn't matter what they do. I'm going to be able to go and be active and find my way. But it can be a little bit intimidating. It can be a little bit. There is some degree of my way or the highway. That's the rationale behind some of it.
Sure.
But overall, they're within. So what I've been told is that within the 6 communities, and there's really not much crossover. There's probably 1 or 2 or maybe 3 that someone like myself might want to join. And within that, they're going to create maybe 2 to 3 different subgroups. And the concept here is a rotating topic, John.
Okay.
So instead of we're kind of going over what we kind of saw there, it'll be much more streamlined. It'll be, hey, For this year, we're going to talk about, I'm making this up, ADCs and immunogenicity, right, or something like that. But there'll be like themes, because what people say is some of the focus groups, they run out of content and they're just spinning for years at a time, and then they kind of ebb and flow, right?
Yep, sure.
So they kind of go back, and there's some that they'll be 2-3 years, they'll publish a paper, and then they'll just fall flat and go away. And so to kind of avoid some of that administrative stuff, right? Maybe there's more behind the scenes here in terms of trying to manage that, phasing it out, funding it. I don't know how it all works, but I think what they're really looking for is it'll be a much more broader. Remember, sometimes a call will have maybe 30 to maybe high watermark is 80. That's what I've seen. I haven't seen too many. Our group usually averages it ebbs and flows. Sometimes it's 40, and we've had up to 60. But rarely is it over 100. Now, some of these calls are going to have like 500 people on it, which on the surface means maybe there is more people to contribute. But in a lot of people I was talking to about it, and a lot is maybe 3 or 4, they were like, man, I'm intimidated in front of 30 people. There's no way in hell I'm going to talk in front of 5.
Yeah, I don't know how
you know what I mean? And like, so it's really, and I think there's still, there's the AAPS people are still trying to, they're going to roll this out, but it's going to be evolving. So big news there. It really is it is something where I'm a little bit saddened that I will be losing, that leadership of the critical reagents, where I'm running up on the 3rd year of leading that. We're going to publish a paper here, hopefully in January or February timeframe, but then it'll cease to exist after that. And so stay tuned on that, John. I don't know. Any thoughts or comments? Otherwise, what else is going on at EBF?
Not really. I'll tell you more about what was going on at EBF. I mean, so it's a little bit in flux, which I will talk about later. But it's obviously I'm there as a smaller molecule person, and although there's still good small molecule presentations, obviously large molecules, it's becoming much more of an emphasis. When I say oligos, there wasn't like major sessions on that. There was a lot of focus on hybrid LCMS, particularly talking about ADCs. So it's like there was at least one ADC session, and they're having a workshop in Malaga next June to discuss ADCs. So because of the complexity of them as analytical species, they're becoming more. There's a little bit more focus on them.
I'll go to Malaga, John. Right? That's like that's one of the nicest places on the planet, if memory serves me.
Yes,
right? Malaga's beautiful.
It is a nice city, and the workshops are good, because I did one on oligos a couple of years ago. And given our focus on ADCs, that's something we should be talking about within a group if someone goes or not. Anyway, that's something to sort of have on the diary. Interestingly, there was a full session on PCR. And also Flow had a session for the first time. So, EBF's obviously looking at other technologies that are complementary to what we've traditionally been talking about at the conference. So that's that was nice. That was good to see. Right. So you might find the attendance changes slightly in terms of the people who are there. I mean, it was on the whole, it was as usual. It was a full. The part of it is they've changed now from a 2.5-day meeting to a 3-day meeting, but you still felt as, even with the 3 days, you were pretty full on for the full 3 days. So it didn't feel like any respite from the 2.5,
Yeah.
But it was good. The vendor exhibition. It slightly changed. So to accommodate because now they were running for majority of the meeting, they're running 3 parallel sessions. So like the amphitheater is with the biggest. They try and anticipate the biggest audience, and then they had one big room, and so then they've actually cut back the main exhibition space to then pulled together a third room for that third session. It meant then that the exhibition space got a little bit jangled around to accommodate that change in room space. So, but again, I think from us, we were still in the main room, but we got a good flow of traffic and certainly a good engagement. And I think that there will be things come from the EBF meeting.
Yeah, same here. I mean, it was John. I think the first one I went to was 2009 seems to be right. But prior to joining KCAS Bio, I went to a couple, and it was in Atlanta, and I think it had like 8,000 people at it.
Sure.
I was like, oh my God, this is huge. And
Yep.
someone's like, no, this is nothing. It used to be 12, right?
Yeah, sure.
And so the meeting was about 3,000, I think was what I heard, 3,500. Not tiny, but not as big as it once was. And, it was interesting because we had a fair amount of just meetings. So I was on the fly a lot, meaning, breakfast, lunch, dinner.
Sure. Yeah.
And even some coffee breaks in there. So it kept me busy. And then in terms of like we were kind of smack dab in the middle of the hall, nothing good or bad, but we were near some of the stages and that was nice. So, and Don presented in one of them. And so, foot traffic was, I guess I would kind of put it like in a slow bucket until Wednesday. And then we got thumped.
Really? And that's exactly the reverse of what you'd expect.
Exactly. I think it was because some of the bioanalytical stuff was later in the week. And of course, that was when it was just Don and I at the booth. So that was interesting. and maybe it was because it was Don and I at the booth. I don't know. But there was a fair amount of people coming by, but there's still a lot of who's who amongst pharma there.
Sure. Yeah.
It was overall, I'd say it was a pretty decent conference and one that we'll continue to go to annually. I mean, it certainly keeps you up to date and good to be seen.
Sure. Absolutely. Yeah.
There was something else on.
I could talk about, KCAS. One thing that, we had a social event on the Tuesday night,
nice.
Which was actually, it was a tour of La Pedrera, which is a Gaudi designed apartment complex. So you meet with a guide who walks you through the mainly the hall and stairs, and shows you some of the design innovations of Gaudi's approach to building an apartment. It's not really a block of apartments, let's call it that. And then you go into an apartment that's furnished as it would be at the time, and then we ended up on the roof with a nice view out to La Sagrada Familia. And then down the stairs to finish with a couple of glasses of cava and some nibbles. That was nice. We had a reasonable group of people to sort of share that with. And then the SciEx event this year, which is always a good event, was actually at a winery outside of Barcelona. So we were driven out half an hour out there. Got to taste a variety of wines, and then you had a mixture with wines and food after that. So Ferran Sanchez, who we've collaborated with on setting up webinars between KCAS and SciEx, he's moved to a more global role. So now there'll be other people responsible for the SciEx event in the future. So it'll be interesting to see what they do from next year onwards. But socially, it was the usual thing that they have the mixers at the end of, because the sessions run like 9 to 6, and at 6 o'clock they have a cocktail session. I think those were relatively quiet this year, but still it's a good chance to talk to people while everyone's that little bit more relaxed. But yeah, it was all in all, it was a good meeting. In terms of news from EBF, Philip Timmerman, who's basically driven EBF for years, is retiring. So he's now. So he'll be I think I was not in the closeout session. Again, I was catching up on some stuff, and so they're basically splitting scientific oversight into one group, and then the logistical to another team dealing with that. And the scientific direction will be driven by Matt Barfield, who's currently at Roche, and he's obviously been involved with EBF for years, and he was particularly talking on the AI side of things. I didn't see much of him for the rest of the meeting. So yeah, it's a changing of the guard with an EBF. So that would be, again, when you talk about AAPS cutting down on their focus groups, etc. And now your EBF is obviously in flux too. So it's going to be interesting to see how this plays out over the next couple of years.
Yeah. Well, I hope there's no dip there. I'm not.
Oh, no. I don't. Yeah, we hope. I don't think so. I think they probably worked on this. So
I think it's getting so big now. I imagine he needed help anyway, right?
Oh, sure. Yeah.
No, that's really interesting.
Been doing this for a long time, because this was what, number 18 of these open symposiums, and obviously they do all the workshops during the year as well. Once you get to a certain age and you want to break and do your own thing, so I assume he's just got to that stage. So yeah, so that I think it was the big closing news, and yeah, it was all in all, it was a great three days. So,
yeah, no, just to round out and wrap up here, and we can be on our merry way. But so I mentioned we had a nice dinner as the biomarker group, and then I stayed. Wednesday was the conference kind of ended. Wednesday there was a little bit of a talk Thursday morning, but kind of Wednesday the exhibit hall wrapped up in the afternoon. But we took a client to see the San Antonio Spurs play the Golden State Warriors, which was a lot of fun. Maybe not as adventurous as what you did. It doesn't sound quite as entertaining and as cultural as what you did in Barcelona, but Steph Curry did score 44 points, which was pretty damn cool. So that was a fun event on our end in San Antonio. And then, of course, you do the river walk, which I don't know if you've been down there.
I've been down there a couple of times.
It's nice. It's very convenient is the word that comes to mind.
Sure.
And had a prickly pear margarita or two, and those were fun, and enjoyed a couple Southwest dishes as well. John, glad that this travel season's behind us. It's been a little bit hectic, I'm sure, for both of us. We've been traveling quite a bit, and I know you're still on the go. You're not even home yet, are you?
No, I fly tomorrow, and then I'm actually getting into Philadelphia because with the whole shutdown, I decided to drive to Philadelphia rather than risk not making a connecting flight. So I'm basically going to drive into Philadelphia and then just stay overnight and then drive home on Thanksgiving morning.
I think you're gonna. I know the shutdown's over, but there is a lot of travel going on, and
I left in snow and sleet when I on the 11th, and then obviously did Edinburgh, etc. And then my wife is actually home now, so like I say, we had a few days in Edinburgh, then I did Barcelona. And then this last weekend, got together with friends from school and got to sample some very, very, very collectible whiskies that are no longer collectible because we drank them all. But I had an introduction to Bowmore 18-year.
Oh, wow.
A Japanese 18-year-old whiskey, which was lovely, and another rare Auchentoshan whiskey. So yeah, it was, and it was the pleasure of getting together with three friends who I haven't seen for a couple of years. So that was it was totally worth having the extra couple of days to catch up with them. That was really good. So
sounds like a great time. I mean, I think I don't know. I can't tell if we kind of went into what our weekend plans were there, but I think you kind of recapped where you got a busy Thanksgiving coming up. It sounds like you're.
I think my Thanksgiving is largely going to be recovering from last weekend, etc. I don't have any major weekend plans at all. It will be just getting home and chilling for a few days before starting in December. But yeah, it's I don't have a lot to add. I'm glad we caught up on this. I like this as a topic actually, and we'll remember it for the future. But yeah, like I say, I'm always happy to travel, but equally I'm always happy to get home after traveling.
Oh,
So
yeah.
I'm at a stage where I'm actually looking forward to getting back and relaxing.
I wonder if this is the first time we've ever recorded on a Tuesday morning, which is due to the holiday and your travel a little bit, but that's a little bit unique.
Sure.
But for me, been really trying to get into both. Thanksgiving's late this year, John, so it feels
Sure.
like Thanksgiving and Christmas are colliding at the same time. So spent some time putting lights up on the house, and then well, the couple weekends ago, my son had his father-son weekend, so I spent a couple nights up in Lawrence. And that was, boy, I am not as young as I once was. I cannot keep up with 20-year-olds anymore, if you know what I mean. And we had a
sounds of things you can't keep up with 60-year-olds either. Judging by the amount my friends and I were putting back.
No, it was a lot of fun, and this upcoming weekend. So we are. This afternoon we're gonna go. So I'm off this afternoon and tomorrow, and then obviously Thursday and Friday in the week.
Sure. Yeah.
We're gonna go surprise our in-laws. My wife's family's coming out there. But they're staying at a little resort about an hour away, and my wife and I are gonna surprise them there for my mother-in-law's 75th birthday. And so we'll spend a night there, and then we'll come home and have a wonderful. My son is home. He came home from KU. He's halfway through his sophomore year there, doing really well in the pre-med dental. He wants to be a dentist. And so we're happy to have him home, and life is pretty good right now, John. But this weekend we'll have a very, kind of just the five of us for Thanksgiving, which is fabulous. It's one of my favorite days of the year, John. I get to do nothing and eat a lot and watch football. But and then over the weekend, I'm not really sure what we got going on because the next few days keeps us busy. But that's it in a nutshell, John. So I'm glad safe travels back. It was.
Thanks very much.
I don't know if you have anything in closing, but it was a fascinating to hear about EBF, and certainly lots of. I think the bombshells are the AAPS communities are going away, and that Philippe is leaving EBF. Boy, that was a little bit of a bombshell to hear.
Exactly. Yep. So,
all right. Well, take care, John.
Yeah. So, thanks very much for this. Have a great Thanksgiving, and we'll talk soon.
Yeah. Happy Thanksgiving to you. See you in the funny page.